Hello, my name is Cody Anderson. I'm a graduate student at North Dakota State University, and I will be talking with you today about a project completed as part of my graduate research. My talk is titled Evaluation of Evolutionary Naivety for 3 Western Fish Species. So first, I like to begin with a thank you for everyone who helped make this project possible. Without your support, this would have been much more difficult. So we appreciate that everything that you all did to make this work happen. So thank you. I want to begin with talking about the evolutionary naivety hypothesis, which has been described by Cox and Lima, 2006. So prey naivety is the ineffective anti-predator defenses owing to the lack of evolutionary history with the given predator archetype. So this means that a prey species has no appropriate defense against a certain type of predator. It's unable to recognize a certain type of predator as a threat. And this can be behavioral, physiological, or morphological. So how does this play into desert fishes? Aquatic islands occur in the desert Southwest in terms of habitat. So on the left side of the screen, you can see a map of the water bodies and how much water was in the desert or within the southwestern United States during the Pleistocene, so about 10,000 years ago. Today, there is much less water. So if you look at the pictures on the right side of your screen, you can see the top one is of the boardwalk in Death Valley, very little water, a lot of desert habitat surrounding it. And the 2 lower photos show just isolated pockets where there's not a lot of connectivity or no connectivity between habitats. So in thinking about this, we can ask the question, are desert fishes evolutionarily naive? And we can look at poolfish as an example. So poolfish evolved in the absence of other fish. And research has shown that they cannot co-persist with mosquito fish. So Goodchild and Stockwell, 2016, evaluated poolfish survival in mesocosms in treatments with just poolfish or poolfish and mosquito fish. So if we look at this graph, we can see treatment groups of allopatric poolfish or poolfish with mosquito fish on the x-axis, and then on the y-axis, we see the number of poolfish juveniles produced within that growing season. And we can see that the allopatric poolfish produce many more juveniles within that growing season compared to poolfish that were in a treatment group with mosquito fish. So poolfish in sympatry with mosquito fish produce almost no juveniles. So that got us thinking, and how can we evaluate anti-predator behavior within fish? So fish are normally swimming around within the water quality, they're foraging, or within the water column, they're foraging, and they're up in the water column. But if a predator fish comes in and eats a prey fish and breaks the epithelium, an alarm substance is released from the epithelium, and we're not sure what exactly the chemical constituents are that make it an alarm substance, but this substance is released into the water from that broken epithelium, and subsequently the other prey fishes that were actively foraging change their activity levels, so they reduce their activity, and they also reduce their position within the water column, and this is a tactic to avoid that actively foraging predator. So what does this look like graphically then? How are we going to evaluate data, and what do we expect? So there's 2 ways to demonstrate anti-predator response to alarm cues. And first is that we can look for a significant treatment effect. So we have pre-stimulus behavior on the x-axis, post-stimulus behavior on the y-axis of this graph, and a treatment of water is denoted by the blue line, and a treatment of alarm cue is denoted by the red line. So when there's a significant treatment effect, we see that there's a difference in elevation of these lines. So the alarm cue line is much lower than the water treatment cue line. So this is what we could say is a significant treatment effect, as denoted by an ANCOVA evaluation. Another way to look at the data or is that you could see a significant interaction term. So again, this graph shows pre-stimulus behavior on the x-axis, post-stimulus behavior on the y-axis. Water treatment is denoted by the blue line, and alarm cue is shown by the red line. And we can see that these lines have different slopes. So with the significant interaction term, the rate of change from y to x is not the same for these two groups, but we can still see from this graph that there's a significant interaction. So there's a significant interaction term here. So when poolfish were evaluated for changes in behavior, this is what we saw. So this is data from Snyder 2019, and we can see the pre-Q vertical position on the x-axis, post-Q vertical position on the y-axis. The treatment of water or control is denoted by the blue, and alarm cue treatment group is denoted by the red. And we can see that these lines are pretty well the same. So there was no change in behavior when poolfish were exposed to conspecific alarm cue. So that got us thinking, and we decided to evaluate behavioral responses of 3 different species of pupfish. So we looked at Amargosa river pupfish, or Cyprinodon navidensis amargosae, which can be seen on the top left. We also looked at Shoshone pupfish, Cyprinodon navidensis shoshone, which is top right. And then the red river pupfish were the 3rd species that we looked at. That's Cyprinodon rubrofluviatilis, and that's shown by the bottom picture there. So why did we choose these 3 species? So Cyprinodon rubrofluviatilis, or red river pupfish, we can see highlighted on the phylogeny here as part of the Southern Great Plains or Northern Chihuahuan Desert group within pupfishes. And these red river pupfish have also evolved in complex habitats with and in symmetry with more species of fish. Shoshone pupfish and Amargosa river pupfish are part of the Western group of the phylogeny. And we can see those highlighted at the bottom of the page here. And they evolved in more simple environments. So Cyprinodon navidensis amargosae, or the Amargosa river pupfish, co-persists with Amargosa river dace, and then Shoshone pupfish are found in Shoshone Spring, and they evolved in allopatry. So on a map, this is where we can see these groups are from very different locations. So Shoshone pupfish and Amargosa river pupfish are found outside of Death Valley or very near to Death Valley, and red river pupfish are found in the Red River Basin in Texas. So how do we go about evaluating these changes in behavior and get the data that we need to see what's going on behaviorally? So we have a fish tank here. We start out with 1 individual fish in a 37-liter aquarium. So that's your standard 10-gallon fish tank. I want to point out the grid line that's drawn on the front of the tank displayed on the page here. So the grid line, or the grid, is what we use to quantify both vertical position in the water column and also activity levels. So the grid divides the tank into 5 levels of the water column, and we can see, we evaluate where the fish is at. Is it in the lowest level, 2nd, 3rd, 4th, 5th level within that tank? And then we also use this grid to figure out how active that fish is. So we use the fish's eye as a focal point, and every time that fish's eye crosses one of those grid lines, that's counted for activity. So when we put the focal fish in this tank, we let it acclimate to the aquarium for 24 hours before trials. And then we observe behavior for 5 minutes pre-treatment, and we record vertical and activity data. And then we deliver a treatment to the tank via a syringe and tubing. So you can see the syringe over here on the right side of the page, and it's attached to tubing. The tubing goes up and into the tank and delivers that treatment right next to the bubbler so that the treatment can be disguised in its delivery to the tank. So the treatment consists of, for alarm cue, 1 centimeter squared of skin per 10 milliliters of DI water, and the control is just 10 milliliters of DI water. And then finally, after the treatment is administered, we record vertical and activity data for 5 minutes on for a 5-minute observation period post-treatment. So this is what it looks like in the lab. We have our camera set up to be recording these trials as we complete them. So we write down vertical activity in real time, and then we score activity data from the videos. You can see the tubing here that comes up and then around to the back of the tank and into the bubbler to deliver the treatment. There's the grid line on the front of the tank. And I also want to point out that there's dividers on each side of the tank so that fish can't see other trial fish. So it's isolated to just that one fish in one tank. So for results, for Amargosa River pupfish, we found that there's a significant change in vertical position. So on this graph, we have pre-vertical position across the x-axis, post-vertical position across the y-axis. And we have the treatment group of alarm cue denoted by the red line and water denoted by the blue line. And we can see that there's a significant interaction term here, or there's a significant interaction effect because these lines have very different slopes. Interestingly, though, there was no significant change for Amargosa River pupfish change in activity. So we have pre-activity across the x-axis, post-activity across the y-axis. The treatment group of water is denoted by the blue line, and the alarm cue treatment group is denoted by the red line. And we can see that these lines look very similar, and statistical analysis for an ANCOVA show that there's no significant change. When we looked at Shoshone pupfish, vertical position, we found that there's a significant interaction term. So we have pre-vertical position across the x-axis, post-vertical position across the y-axis. Again, the alarm cue treatment group is denoted by the red line, and the water treatment group is denoted by the blue line. And we can see the slopes of these lines are very different, indicating a significant interaction term. Shoshone pupfish also showed a significant change in activity. So we have pre-activity across the x-axis, post-activity across the y-axis. Alarm cue is denoted in red. The treatment group of water is denoted by blue. And we can see that there's a significant interaction term here because the two lines, again, have very different slopes. So we're seeing a reduction in activity based on the treatment of alarm cue. When looking at Red River pupfish, we found a significant change in vertical position as far as the interaction term. So we have pre-vertical across the x-axis, post-vertical across the y-axis. Alarm cue is denoted by red. The treatment group of water is denoted by blue. And we can see that these lines have different slopes, indicating a significant interaction term. And finally, Red River pupfish also showed a significant change in activity. So for preliminary analysis here, we can see the two lines indicating very different slopes. So we have pre-activity across the x-axis, post-activity across the y-axis. The treatment group of water is shown by the blue line, and the treatment group of alarm cue is shown by the red. And we can see that these lines have very different slopes, and analysis indicated a significant interaction term. So in summary, Red River pupfish, Shoshone pupfish, and Amargosa River pupfish all show a behavioral response to conspecific alarm cue. So we all saw changes in behavior for the treatment groups that were given alarm cue. These findings are not consistent with the predator naivety hypothesis. And future work, so potentially with white sands pupfish or maybe Pecos pupfish, should include pupfishes that evolved in isolation for longer periods of time to get a good look across all the pupfish groups. So thank you very much for listening to my talk.